Imagine for a second that you spend your entire weekend weeding a garden. You know, you're out there on your hands and knees, pulling up roots, meticulously making sure every single visible weed is gone.
Right, getting the soil looking completely pristine.
Exactly. You wash your hands, you're feeling great about the hard work. But then like a few months later, you walk outside and those exact same weeds have sprouted back. And they're somehow thicker, more deeply entrenched and way more stubborn than before.
Yeah, which is just an incredibly defeating feeling, to know you did everything required to clear that space, only to realize that the environment beneath the soil was already quietly primed to bring those weeds right back to life.
And, while that's just an annoyance when you're dealing with a backyard garden, it's actually a tragic and brutal reality in oncology.
It really is.
Specifically, we're talking about hepatocellular carcinoma today, or HCC,
right.
Which, for those of you listening who might not be super familiar with that term, it's the most common type of liver cancer.
Yeah, and the statistics surrounding HCC are, well, they're sobering. The 5-year survival rate sits below 20%.
Wow. Under 20%.
Yeah, and a massive reason for that really low number is recurrence. Because even when patients undergo these aggressive, highly targeted treatments, and even when their scans come back looking completely clear, the cancer just frequently finds a way to roar back.
Which is the entire focus of our deep dive today. We're looking at this highly elusive state called minimal residual disease, or MRD.
Right, which refers to the tiny, tiny fraction of tumor cells that somehow survives the initial bombardment of cancer therapy.
And these surviving cells are often so few in number that they're entirely undetectable on standard radiological scans, right?
Right, exactly. They go into hiding deep within the tissue, lying totally dormant for months or even years, only to eventually trigger a massive recurrence.
So to figure out how this happens, we are unpacking a truly groundbreaking study published in Nature Cancer. The researchers here, they essentially set out on a mission to figure out exactly how these specific cells pull off this vanishing act.
And they focused on patients who underwent a procedure called transarterial chemoembolization, or TACE.
Right, TACE. And to understand why this paper is so important, you really have to understand what TACE actually does to the liver.
It's intense. Doctors basically thread a catheter directly into the blood vessels that feed the liver tumor.
They pump high doses of chemotherapy straight into the mass, and then, and this is the wild part, they deliberately block the blood vessel to choke off the tumor's oxygen supply.
Yep, it's heavy duty. Localized chemical warfare and starvation all happening at once. Yet, inexplicably, a tiny fraction of cells manages to survive this apocalyptic event.
It's crazy. And, historically, oncologists and researchers assumed those surviving cells were just like genetic anomalies.
Right. The thought was that they were super mutants naturally immune to the drugs, or that they rapidly mutated during the treatment to develop resistance.
But this study completely upends that assumption. It proves that the survival of these cancer cells is so much more insidious than just individual genetic toughness.
It's not just about the cells themselves. It's about the highly organized, microscopic neighborhoods they build to actively protect themselves from the therapy.
And the immune system, right. Which brings us to the technology they use to figure this out, because I am just fascinated by this part.
Oh, the tech is incredible.
They didn't just catalog what cells were left floating around in the liver. They looked at exactly where those cells were living in relation to one another. It's essentially microscopic urban planning.
Yeah, before we get into how the cancer survives, we definitely have to look at how these researchers managed to map this microscopic battlefield. Because to truly understand cancer survival, you cannot just look at a tumor as a giant, uniform lump of bad tissue.
Right.
You have to understand its microenvironment. The researchers took a massive leap forward in spatial biology here, moving from the macro problem of a visible liver tumor down to the cell-to-cell, micrometer-by-micrometer interactions.
Okay, let's unpack this for a second. Because in the past, if you wanted to analyze a tumor to see what genes were active or what cells were present, you'd take a biopsy, break down the tissue, and sequence it.
Which is standard, yeah.
But I always think of that traditional method like taking a beautifully baked fruit tart and throwing it into a blender to make a smoothie.
That's a great way to put it.
Like when you test that smoothie, you can easily identify the ingredients. You can say, oh, there are strawberries, blueberries, flour, and sugar in the mix. But you have absolutely no idea how that tart was originally constructed.
Exactly. You lose the fact that the strawberries were arranged in an intricate circle on top of the cream, and the blueberries were baked into the crust.
You completely lose the architecture.
Right, and traditional bulk sequencing destroys the spatial context, which turns out to be the most important part of the story.
Hmm.
So, to avoid making a smoothie, the researchers in this study used a cutting-edge technology called Codex.
Which stands for code detection by indexing, right?
Yes. Codex provides a high-resolution, wildly detailed satellite map of the cellular city. It allows scientists to see exactly which cell lives next door to which cell without destroying the structural integrity of the tissue slide.
And the mechanics of how Codex works are just mind-blowing. Like, they analyze tissue from 108 human HCC patients.
Yeah, 108 patients.
And they compared primary HCC, meaning tumors that were freshly diagnosed and hadn't been treated yet, with residual HCC, which are the stubborn tumors that survived the PACE therapy.
And to map this out, they used a panel of 41 oligo-tagged antibodies to analyze a staggering 1.07 million individual cells.
Over 1 million cells. But wait, how do you look at 41 different things on a single slide without it just becoming a blurry mess of colors?
The secret is DNA barcoding. See, in standard imaging, you might use 3 or 4 fluorescent colors to highlight different proteins, but you quickly run out of distinct colors.
Right, obviously.
So with Codex, they attach a unique synthetic DNA barcode to 41 different antibodies. These antibodies are designed to latch onto specific proteins on the surface of immune cells, structural cells, and cancer cells. The machine washes fluorescent probes over the slide that bind to just one specific barcode, takes a picture, chemically washes the color away, and then moves to the next barcode.
Wow. So by stacking these dozens of images on top of each other, they can definitively identify the exact type and state of over 1 million individual cells while keeping the neighborhood entirely intact.
Exactly. It's a perfect map.
So they generate this incredibly detailed satellite map of 1 million cells, and then they compare the untreated primary tumors to the residual tumors that survived the chemoembolization. And I guess the obvious question is, what changes after the bombs drop?
Well, what's fascinating here is that the fundamental demographic makeup of the tissue is completely altered. The residual tumor is not simply a miniaturized, battered version of the primary tumor.
The treatment reshaped the entire landscape.
Completely. Normal cells you would expect to see in a liver environment, like everyday neutrophils and structural fibroblasts, were severely depleted.
And what replaced them?
In their place, the residual tumors were heavily enriched with immunosuppressive cells. There were massive spikes in PD-L1 positive macrophages, mast cells, and exhausted CD8 positive T cells.
Man, it sounds like the therapy wiped out all the regular civilians in the city, and suddenly the streets are swarming with corrupt security guards and completely exhausted soldiers.
That is exactly what it looks like on the map.
Let's dig into those T cells for a second. Because in a healthy body, CD8 positive T cells are the good guys, right? They're the executioners of the immune system, designed to hunt down and kill mutated or infected cells.
They are the absolute front line of defense against cancer. But in the residual HCC tissue, these specific T cells were found in a state of exhaustion.
And exhaustion in cellular biology isn't just a metaphor for being tired, is it?
No, not at all. It is a measurable physical state. These T cells express high levels of specific surface markers, primarily PD-1 and TIM3.
Okay, and what do those markers mean?
When a T cell is plastered with these markers, it is physically present in the tumor bed, but it is functionally deactivated. It is basically chemically lobotomized.
Oh, wow. So it can bump right into a cancer cell, but it cannot fire its weapons or sound the alarm?
Exactly. It's useless.
It is terrifying to think that right now in our bodies, we might have fully capable immune cells that have been chemically hypnotized into doing nothing while a threat is standing right in front of them.
Yeah, it's a sobering thought.
And then there are the macrophages. I always learned that macrophages were sort of the Pac-Men of the immune system. You know, they patrol the body and engulfing and eating cellular debris, dead tissue, and pathogens. But you mentioned these macrophages are marked with PD-L1. What does that specific marker do?
So PD-L1 acts as a molecular stop sign. It is an immune checkpoint protein. When a cell displays PD-L1 on its surface, it sends a direct signal to the PD-1 receptors on those T cells we just talked about.
Telling them what?
Telling them, nothing to see here, stand down, do not attack.
Ah, okay.
And many types of aggressive cancer cells mutate to display this stop sign themselves to evade the immune system. But in this residual tumor environment, the cancer isn't holding the stop sign.
Let me guess. The macrophages are holding it.
Yes. The very immune cells that should be cleaning up the disease are the ones holding up the PD-L1 stop sign to protect the area.
That is so devious. So if we do a quick recap here, we have our demographics. We know the million cells on our slide are dominated by these immunosuppressive macrophages and exhausted T cells.
Right.
But just being in the same general area of the liver isn't enough to protect a tumor. Like, a corrupt security guard is completely useless if he's standing 3 miles away from the bank he is supposed to be protecting.
Exactly. The researchers realized they needed to measure the exact distances between these cells to see who was interacting with whom.
So they moved from demographics to spatial interactions.
Yeah. The researchers fed this massive spatial data set into a clustering algorithm. They programmed the algorithm to look for distinct patterns of proximity-like, which cells consistently clumped together across all 108 patients.
And what did the algorithm find?
The algorithm identified 9 specific recurring cellular neighborhoods within these liver tumors.
9 distinct microscopic zip codes.
Yes. And when they zeroed in on the residual tumors that survived the TACE treatment, one neighborhood in particular had completely taken over the landscape, while the neighborhoods filled with active cancer-fighting cells had virtually disappeared.
And what was that dominant neighborhood?
The dominant surviving structure was something they termed the M2-like macrophage immune neighborhood. It basically became the defining feature of the cancer's survival strategy.
The paper makes a really big distinction about these being M2-like macrophages. What is the difference between an M1 and an M2 macrophage? And why does the cancer prefer the M2 variety?
Well, macrophages generally polarize into 2 distinct functional states based on what the body needs at the time. M1 macrophages are aggressive and inflammatory. They're the soldiers that attack active infections and destroy diseased tissue.
Okay, so M1 equals soldiers.
Right. M2 macrophages, on the other hand, are the medics and construction workers. Their job is wound healing, tissue repair, and basically turning off the inflammatory response once an infection is cleared.
Oh, I see where this is going.
Yeah. The chemotherapy from the TACE procedure causes massive tissue damage. The cancer expertly exploits this by recruiting M2 macrophages, tricking them into treating the chemo damage as a standard wound that needs to be healed and protected.
That's incredibly smart. And the researchers drew very strict boundaries to prove these cells were working together, right?
They did. They defined direct contact as cells touching within a 25 micrometer radius. They also tracked indirect contact within a 25 to 100 micrometer radius.
And what did they see within that radius?
They found that very specific cancer cells, stem-like tumor cells, identified by the proteins EpCAM and CK19, were deliberately, physically huddling together with these M2-like PD-L1 positive macrophages.
Okay, wait. I need to push back on the timeline of the survival mechanism for a second.
Sure.
Because if the take-off of therapy cuts off the blood supply and dumps a massive localized dose of toxic chemotherapy directly onto the tumor, how are these stem-like cells surviving the initial blast to even build this neighborhood?
That's the million-dollar question.
Right. I mean, the M2 macrophages can't shield them if the cancer cells are already dead from the first wave of treatment.
Well, that is the defining characteristic of cancer stem cells. The presence of those specific proteins we mentioned, EpCAM and CK19, is a crucial detail. Those proteins are markers of a stem-like state.
Okay.
Unlike standard fully mature tumor cells that make up the bulk of a mass, cancer stem cells are an immature, highly adaptable subpopulation. They are essentially the seeds of the tumor.
And how does being a seed help them survive TACE?
They naturally possess a very slow metabolism, and they're uniquely adapted to survive in hypoxia, which is a severe lack of oxygen.
Ah, and TACE is designed to cause hypoxia by blocking the artery.
Exactly. Furthermore, traditional chemotherapy relies on targeting cells that are actively and rapidly dividing. Because these stem-like cells are dividing so slowly, the initial chemoblast essentially just bounces off them.
Oh, wow. So the fast-growing bulk of the tumor dies off, but these slow, sleepy stem cells weather the storm.
Yes. However, they know they cannot survive the immune system's secondary cleanup crew without help, and that is when they pull the macrophages in close.
Okay, so let's synthesize this. The stem cells survive the blast, and then they huddle up with the M2 macrophages. The macrophages throw up that PD-L1 stop sign, which directly interacts with the nearby CD8 positive T cells, forcing them into that exhausted, deactivated state.
You got it.
It creates a localized microenvironment where the immune system is completely paralyzed.
It is a perfectly coordinated physical barrier. But you have to remember, proximity and physical contact are only half the story. Cells don't have voices. They communicate and exert control over their environment by secreting chemicals.
Right, so the researchers needed to move from a spatial map to a chemical map to understand how this neighborhood was maintaining its control.
Exactly.
And to figure out the chemical signals, the research team used another cutting-edge platform called spatial transcriptomics, specifically the nanostring geomex technology.
Yes, nanostring geomex.
If the codex technology was the satellite map showing us exactly where the bad guy's hideout is located, spatial transcriptomics is kind of like sneaking in and wiretapping the building to hear exactly what they are whispering to each other.
That is a highly accurate way to visualize it. Spatial transcriptomics allows researchers to select incredibly specific microscopic regions of the tissue slide.
Like down to the micrometer.
Yeah, in this case, they zeroed in on the exact 25 micrometer interface where the stem-like tumor cells and the macrophages were touching. They used targeted beams of UV light to cleave off tags from the messenger RNA exclusively in that tiny illuminated circle.
Wow.
This tells them exactly which genes are being turned on and off and what proteins are being manufactured right there at the exact point of contact.
And here is where it gets really interesting, because when they wiretapped these specific residual neighborhoods, they uncovered a massive upregulation of one particular chemical pathway, TGF-beta, or transforming growth factor beta.
Yeah, TGF-beta, it's a cytokine, which is a signaling protein.
And what does it normally do?
Well, in a healthy human body, it plays a vital role in controlling cell growth, guiding embryonic development, and suppressing severe inflammation. But inside this residual tumor neighborhood, TGF-beta is entirely weaponized by the cancer to ensure its survival.
The transcriptomics revealed that the M2 macrophages are the ones primarily manufacturing and pumping out a specific variant, TGF-beta1. And they are feeding this chemical directly to the stem-like tumor cells they are protecting. And the effect it has on the cancer cells is just devious. It forces them into a state of deep, persistent dormancy.
And this chemical induction of dormancy is the ultimate survival mechanism. Because, as we discussed, chemotherapy is designed to seek out and destroy cells that are actively going through the cell cycle and multiplying.
It makes me think of chemotherapy as like a motion sensor weapon, or honestly like the T-Rex in Jurassic Park. It can only see you if you are moving.
That's a perfect analogy.
By constantly spraying the stem-like tumor cells with TGF-beta1, the macrophages are forcing the cancer cells to stand perfectly still. They stop dividing entirely.
So the chemotherapy sweeps right through the liver, but it doesn't attack the stem cells because it doesn't recognize them as an active threat.
The cancer is using the body's own immune cells to create a localized chemical force field that induces a state of hibernation.
Exactly. And if we connect this to the bigger picture, while the cancer cells are sleeping safely, the macrophages are actively remodeling the environment around them.
How so?
Well, the spatial transcriptomics also showed that these macrophages were upregulating pathways for angiogenesis.
Meaning they were sending signals to build new blood vessels to supply the area with fresh nutrients.
Right. They were also upregulating invasiveness pathways. They are literally paving the roads and laying the physiological groundwork for the cancer to wake up and dramatically spread when the environment is safe again.
Okay, we need a quick summary of this incredibly complex sequence of events because there's a lot here.
That's a lot, yeah.
So the chemotherapy hits the liver. The fast-growing cancer cells die. But a few slow metabolism, stem-like cancer cells survive the blast. They immediately recruit M2-like macrophages and physically huddle together.
Check.
The macrophages display a PD-L1 stop sign to exhaust any attacking T cells, while simultaneously spraying the area with TGF-beta to put the cancer cells into a chemical hibernation. They build a bunker, lock the doors, and wait.
That is exactly what is happening.
So the researchers mapped the entire defense system. But a map is only useful if it helps you win the war. How do we use this knowledge to actually cure the patient?
Well, to test potential cures based on this new map, the researchers had to transition from human clinical observation into preclinical therapeutic testing. You can't just blindly test unproven combination therapies on human patients.
Obviously not.
So instead, they utilized 3 different, highly sophisticated mouse models to recreate this exact state of minimal residual disease.
And I should note, these were not just standard laboratory mice. One was an orthotopic doxorubicin-resistant model.
Right, which sounds like a mouthful.
It does. Without getting lost in the weeds of the terminology, orthotopic basically means the researchers didn't just inject human cancer cells under the skin of a mouse, which is common but not very realistic.
Yeah, that doesn't accurately mimic the liver environment.
Right. They actually grew the tumor inside the mouse's liver in the anatomically correct location. Then they treated the mouse with the chemotherapy drug doxorubicin to perfectly mimic the environment and tissue damage a human patient experiences during TAEs.
And they also used a transgenic MYC-HCC model to ensure their findings held true across different genetic drivers of liver cancer.
Okay, so they have these super accurate mouse models.
Yeah. And once they confirmed that these mice had developed the exact same M2 macrophage and stem cell neighborhoods we saw in the human patients, they initiated their attack.
And what was the attack?
They used a combined blockade. They hit the neighborhood with an anti-PD-L1 drug and an anti-TGFβ drug at the exact same time.
Okay, I really want to clarify why they needed both drugs.
Why?
Because PD-L1 immunotherapy is already a blockbuster treatment in oncology, right?
No, absolutely.
We hear about it constantly in the news as a miracle drug for certain cancers. Why wasn't the standard PD-L1 blockade enough to clear out these residual cells?
Because the tumor neighborhood has evolved a brilliant fail-safe mechanism. If you only administer an anti-PD-L1 drug, you are successfully removing the molecular stop sign from the macrophages. That is a great first step, and it does wake up the exhausted T cells.
Okay, so the T cells are awake.
Yes, however, if that localized TGF-β force field is still actively being pumped into the tissue, the environment remains deeply immunosuppressive, and the cancer cells are still locked in their protected dormant state.
It's like trying to raid a highly secure bank vault. Administering the PD-L1 drug is like taking out the security cameras and disarming the guards.
Okay.
That's necessary. But if the giant 2-foot-thick steel vault door of TGF-β is still locked shut, you still can't get to the money. You have to execute both steps simultaneously.
That is the perfect way to visualize the dual blockade. You have to drop the chemical shield by blocking TGF-β, which forces the cancer cells out of dormancy, while simultaneously disarming the guards by blocking PD-L1.
And what happened when they did both?
When the researchers executed this combined therapy in the mouse models, the physical structure of the tumor microenvironment completely collapsed.
Wow. And the codex mapping showed that the therapy literally physically excluded those bad immunosuppressive macrophages from the neighborhood.
Yep, they were gone.
Because the chemical shield was down and the stop signs were ripped away, massive waves of fresh, activated CD8 positive T cells were able to flood directly into the tumor bed.
And those activated T cells finally had clear targets. They successfully hunted down and completely eliminated the residual stem-like tumor cells.
That is incredible.
By dismantling the neighborhood rather than just poisoning the cells, they prevented the recurrence of the cancer entirely in these models.
So if you're listening to this, you might be wondering what this deep dive into liver cancer microenvironments means for the broader future of medicine.
It represents a monumental paradigm shift in how we view oncology.
It really does. For decades, the prevailing strategy for treating cancer was to simply develop more and more toxic drugs, hoping to kill the cancer cells slightly faster than the drugs killed the patient.
Right, which is a brutal race to win.
But this paper proves that the future of survival is actually about microscopic urban planning. It is about destroying the specific neighborhoods and supply lines that allow the cancer to hide in the first place.
And the implications of this approach extend far beyond just hepatocellular carcinoma. This raises an important question, right? If scientists can successfully target these spatial interactions to eliminate minimal residual disease in the liver, this dual blockade strategy could theoretically revolutionize adjuvant therapies for almost all solid tumors that face high recurrence rates.
Which are the treatments given after a primary surgery or chemo, right?
Exactly. Breast cancer, colon cancer, or lung cancer, they all struggle with dormant residual cells waking up years later.
So we are looking at a totally new way to navigate the battlefield. You aren't just shooting at the enemy hoping you hit them all. You are dismantling their bunkers, turning off their communication lines, and relying on the patient's own naturally evolved immune system to sweep the area clear.
It is an incredibly elegant and targeted solution to one of the most brutally complex biological problems we face. It shifts the power dynamic back to the immune system.
We started this conversation by talking about the deep frustration of weeding a garden perfectly, only to have the roots survive underground and sprout back when you least expect it.
Yeah.
We have now seen exactly how cancer builds an intricate chemical root system using our own immune cells as a shield to guarantee its inevitable return.
It's sobering, but also hopeful now that we can map it.
Definitely. But I want to leave you with one final, slightly unnerving thought to mull over.
Oh.
If specialized cancer cells can form these highly coordinated, perfectly balanced protective neighborhoods to survive extreme chemical warfare like chemotherapy, what other dormant conditions or viral diseases in our bodies are just hiding in the right microscopic zip codes surrounded by unwitting cellular guards, simply waiting for the perfect chemical signal to wake up?
Man, it absolutely forces you to look at the human body not just as a single organism, but as a vast collection of deeply complex, highly communicative, and sometimes treacherous neighborhoods.
It really does make you rethink everything going on beneath the surface.